So let's introduce Professor Jacques Tuszynski. Well, I will do a summary because his CV is 79 page, so it's all about massive contributions. So it's not easy. He's head of the Division of Experimental Oncology at the Cross Cancer Institute in Edmonton, Alberta, Canada, and also full professor at the Department of Physics of the University of Alberta, also in Edmonton. And he has been teaching in Poland, in China, in Belgium, in France, in Denmark, in Germany, in several places in Canada. He's now opening a group in Italy, I think, starting a group in Italy. So he is active in physics of condensed matter, chemistry, biophysics, nanotechnology, and oncology as well. He presented his master in 80 on theoric and magnetic phase transition at the University of Poznan in Poland, and its PhD in 83 on theory of something about spectroscopy, no? At the physics department of the University of Calgary. This is in Canada. He was beneficiated by more than 90 grants, and he received many distinctions. One of them is that I noted especially is that it has been selected by the Swedish Academy of Science as a nominator for a Nobel Prize in Physics 2009, 2012, 2015. He has also participated in the organization of 50 conferences. It's all about statistics with Jack. He has been member from the editorial of, or he is, I've been confused, 17 journal. He taught in oncology, system biology, cancer, phase transition and critical phenomena, superconductivity and superfluidity, statistical mechanics, solid state physics, condensed matter physics, quantum theory of solids, theoretical biophysics, advanced quantum mechanics, molecular biophysics, electromagnetism, nonlinear differential equation. I think it's worthwhile to note this. He already formed 22 postdoc fellows, 28 PhD graduate students, 14 masters. He has a citation impact of 7,423, and he wrote several books and so on. And he has more than 400 papers actually, 20 patents. Yeah. I think we will have a nice talk this morning. Thank you very much.
Thanks, Igor. Great to be here. No pressure. I feel no pressure after this introduction. By the way, my institute is called the Cross Cancer Institute, and Cruz is cross in English, so there's some connection. And in both cases, these are names of people. Cross was doctor, one of the doctors, oncologists, and the institute is named after him in Edmonton. So I prepared 2 talks because it's 3 hours. It was supposed to be, no, it's 2.5, minus the coffee break. But I'll try to cover 2 aspects. This is systems biology, but I don't want to jump into it right away. And the title of my talk is Systems Biology and Disease. And I'm interested in curing diseases. So it's all about drug discovery, drug development. So the first part of my talk today will be about drug discovery, traditionally and computationally towards computational drug discovery. The second will be how systems biology can optimize drug selection, combination of drugs, and improve outcomes. So it's towards practical applications. So the first part, as I said, is the past, present, and future of drug discovery. I have very optimistic feelings about drug discovery in the future, and I'll try to explain why. So this is to show you it's far away from here, and we're in the middle of our summer, and it's exactly the same temperature as here. You don't want to know about the winter in Edmonton. But this is what it looks like now, quite nice, and this is for you. 5 months of snow guaranteed. You can sell it and bring it to Brazil and make a lot of money. Okay, so the motivation for it is medical errors. The medical practice is highly regarded. It's one of the most noble professions, but it's one of the most backward and underdeveloped activities of the human beings. I mean, 1 in 3 errors in medical practice is documented. It has not moved much since this statistics has been published about 20 years ago. It's still about, it's called 1 sigma. Namely, we are operating in medical practice within 1 sigma. That means exactly 1/3 of the probability of error is 1/3. And maybe half of that is actually serious, life-threatening errors of medical professionals. And some of it is the administration of the drugs, route and choice of drug. And in our institute, we experienced some of that, actual cancer patients dying because they were overdosed on chemotherapy. And by comparison, we're used to electronics, aviation, very safe, and everybody's freaking out about flying, but it's like aviation industry operates at 6 sigma. There is 1 error in a few million of some kind, and of course, these are fatal, but medical errors are also fatal. So how do we improve medical practice? And I'm specifically interested in pharmaceutical development because most of the medical practice is about diagnosis and prescription. So first of all, this will be a very general introduction. So I'm assuming nobody knows much about this, and maybe some people in the audience are pharmaceutical scientists, and maybe you can go to sleep for the next 20 minutes. I'll wake you up. But for those who are not, then maybe this is kind of useful introduction. So a drug is a substance that treats, cures, hopefully cures a disease. In most cases, maintains, stabilizes symptoms. And most of the time, it's a small molecule, and it's a very precise definition of what a small molecule is. Typically, it's under 500 daltons in molecular weight. Interacts with the target. That's another technical term. What's a target? We'll talk about this. Biomolecule, typically protein, enzyme, or DNA, which is a biomarker. It's involved in the pathological changes in disease, and much of the discovery is about identifying targets. We'll talk about how many targets we know and which ones are important. My main interest is oncology, cancer, and we'll talk about that too. But my talk actually is an introduction to the talk of Nicola and Tatiana. Later on, they will go into specifically oncologic applications. And drug discovery is a process of finding such molecules. And we've been doing drug discovery for thousands of years, but most of it has been accidental or anecdotal. And the difference between drug discovery and drug design is that discovery is, as I said, it's typically accidental. Design is rational, intelligent, using some principles. And there are emerging numbers of principles that are important in drug design. So what are the targets? And as of today, roughly 5% of the human genome is considered druggable. That is, defines targets for various diseases, all diseases. So altogether, we can say, as of today, 1,500 proteins and enzymes are targets for various diseases. By the way, for cancer, 1/3 of all targets are relevant for cancer. So about 500 targets exist for cancer. And that already throws a big monkey wrench into the problem. If you have that many targets, and cancer, of course, is not a single disease. It's more than 200 diseases called the same name, and then you specify them and so on. So with that many targets, and I'm kind of hinting at the conclusion of this talk, you need to do something much more than just try to find a one silver bullet that will hit the target and this is it. This will never happen for cancer, in my opinion, my humble opinion. Combinations, cocktails, just like AIDS, HIV, was eventually normalized as a chronic disease because of the cocktails. Cancer will undergo this kind of evolution as well. Cancer research and cancer. Okay, so before I get into computational stuff, I just want to give you a very brief historical overview of pharmaceutical sciences. It goes back actually to ancient Egypt and China, and mostly based on plants. But there's some very interesting historical notes. So Galen is the father of Western pharmacology, practiced in Greece and Rome almost 2,000 years ago. But the main advance, actually, it's a brilliant set of principles were introduced by Avicenna or Ibn Sina. He was Persian, I think, and that's 1,000 years ago. And he formulated principles of pharmacology. Some of these are still valid today, and I bolded some of it. So he formulated drug must be free of any extraneous accidental quality. In other words, purity. You have to purify. That's still one of the cornerstones of Western pharmacology, purity, and it's part of the requirement. The second is must be simple, not must treat as simple, not a composite disease. In other words, if somebody has comorbidities, 3 diseases, you have to treat each separately, and you have to develop a drug. Let's say cardiovascular plus liver disease, hepatitis. People may have diabetes, and all of it together. You have to develop a drug for each separately, and he already recognized this aspect of drug development. And then he also recognized something which is now coming back big time: repurposing. That is, drugs may be active on several diseases, and primary disease may in fact be not the best use of that particular drug. You can recycle for something else. Now, these days, aspirin, anti-inflammatory is recommended, preventative for cardiovascular cancers as well, and everybody knows about Viagra, and things like that. So there's always multiple possibilities. He also defined quality of the drug by the strength or potency. That's going to figure prominently in the rest of my talk. Potency of the drug, which is in modern terms defined as the affinity for the target, chemical affinity. And there is he also formulated concepts of pharmacokinetics. So how many of you know the concept of pharmacokinetics and pharmacodynamics? 2 aspects. Some of you do. So pharmacokinetics is the fate of the drug of the body. In other words, the body is a container of containers, and the drug with the bloodstream flows from container to container and is being eliminated. We'll come to that later on. So that's pharmacokinetics. Pharmacodynamics is the action of the drug once it reaches the target. So he also mentioned Avicenna mentioned the need for repetitive testing of a drug, which is statistical significance. He didn't use these terms, but in modern medicine and pharmacology, statistical significance is of primary importance. Right? You have to validate this statistically. Which leads to clinical trials, experimentation with the human body. And he said, which is actually so very true today, and I'm quoting, experimentation must be done with the human body for testing, because testing on a lion or a horse might not prove anything. And in today's pharmacology, you need to test a drug in animal trials on 3 different species, 2 of which are non-rodent, 1 of which is non-rodent. So it could be mice, could be rats, and the 3rd one, rabbit or pig or dog. Okay. Anyway, and then later on, we move forward to 500 years ago, Paracelsus in current Switzerland, I think, practiced and defined the human body as a chemical laboratory. So we are now already using terms close to this conference, to this workshop, because of the systems, bioreactor, in other words. Okay, and then actually the paths diverge between Western medicine and Eastern medicine. And now there's some kind of tendency to convergence. Eastern medicine and pharmacology are based on natural products of many active ingredients. Western is based on a single silver bullet, single active ingredient to high purity. The problem with Eastern medicine is that there is no quality control by and large. The purity is a big issue. Different sources, different compositions. You know, you can call it ginseng, but it's Korean, it's Siberian, and it's Chinese, and different sources give you different purities, and you don't know what you're doing. It doesn't mean that it's wrong. It requires a little bit of purification and scientific approach. Okay, so then later on, in 19th century, drugs were tested on animals on a systematic basis, and 20th century introduced in vitro testing, which is cell culture, as one of the early stages in preclinical trials. And then, starting from the 1970s, there were introduced methods involving purified proteins. So protein-based assays, and let's say DNA-based assays, single molecule, and theoretical approaches, leading to computational with the advent of supercomputers. And the computational work actually was, in my opinion, premature. It was seriously considered in the 1990s, and then more or less doubt was cast. The computers were not powerful enough, the programs were not predictive enough, and a lot of big players in this field, and I spoke to vice presidents of Pfizer, for example, who said computational drug discovery is making hot air very expensively. In other words, running computers, heating the room, and ending up with nothing, which is not quite true. And now, of course, in the year 2000 and beyond, there is a lot of experimental techniques which are very precise, and in parallel, computational methods have refined the toolbox tremendously. So, but still, and I complained about the errors in medical practice, the practice of drug discovery is, in my opinion, and I challenge the audience to find another example which is as error-prone as drug discovery, and I don't know of any, which is, success rate is 1 in 100,000, optimistically. Maybe 1 in a million. It's really like looking for a needle in a haystack. Success being defined as introducing a new drug into the clinic. And it takes typically, these days, 15 years and more than $1 billion of drug development. How it's spent, it's another topic. I don't want to get into political issues, but and typically in a year, there's only under 10 new drugs introduced into the clinical practice worldwide. 10, 6, sometimes more. And this is the flow chart of drug discovery today, starting from basic research. 50% of all pharmaceutical discoveries start with university or government research, institutes like this one. The other 50%, and diminishing actually, is initiated by big pharma. These days it's all outsourced to academic institutions. The early stage, which is cheap but error prone. And then preclinical testing and going through phase 1, phase 2, phase 3 clinical trials on humans, and eventually approval by agencies such as FDA or Health Canada or the European equivalent, and post-market analysis. It's called phase 4. So preclinical tests involve a lot of aspects, and I'm saying all of this for you to give you the background, because much of it can be computerized. And we trust our cell phones. Everybody now has pretty much access to all the information in the world through the cell phone, and we trust the functioning of the cell phone. Somehow, this is an area where automation and predictability is almost unknown. And I know it from experience because in the past 12 years I've been doing drug discovery within the Cross Cancer Institute. We have one compound in preclinical trials and spent only $5 million, but I thought we could accelerate it, and it's very difficult. Some of it is hard to accept, but because of the need to reproduce many times the same thing, to demonstrate chemical stability, chemical purity, do everything at, let's say, GLP levels, then for humans, GMP levels, and costs escalate incredibly when you do that. I think in the academic setting, we're not used to this kind of stringent quality control, and I understand the need, but I think maybe we are losing it. Just for the sake of stringency, we end up throwing the baby with bathwater and ending up with nothing in the end. So toxicity, efficacy, which is the potency of the drug, pharmacokinetics, which is how it behaves in the human body, initially in animal, because we can't give humans something which has not been tested or proven safe. Scanning for rare events, and that is the big problem. It's very difficult to predict. This is an aspect which is very hard to predict. And pharmacodynamics, additional effect on human physiology, metabolism, things like that. So eventually end up with the investigational new drug, which can be approved for testing on humans. And testing involves acute and chronic toxicity. When you hear the word chronic, it means you have to do a long-term study, and some